Hey everybody, today we're moving on. We're going to take a look at phylogeny and the tree of life. So we're actually shifting focus from how evolution works to the pattern that evolution produces over time. When we talk about phylogeny, what we're looking at is the evolutionary history of a species or a group of related species. And so we look at things like the fossil record, at molecules, and at genomes when we talk about phylogeny. Systematics is the study of biological diversity and classification. And so it uses evidence from the fossil record and from other sources to help reconstruct phylogeny. And so really systematics fuses phylogeny, which is the tracing of evolutionary relationships, as well as taxonomy, which is the identification and classification of species. So taxonomy is really natural to humans. We like to group things kind of accordingly, whether it's how they look or how related they are. And so with taxonomy, we have our scientific names that we give every different type of species. Scientific name is composed of genus and species. It's going to be written in Latin, and it's either shown in italics if it's typed or underlined if you write it by hand. And it's governed by a set of rules and procedures. And so we'll be working with binomial nomenclature. Binomial meaning that you've got two names for each organism. So for example, we are Homo sapiens. And so that is our genus, Homo, and then our specific epithet, which is sapien. And so this is going to sort of show us a hierarchical classification system that arranges life into groups. Now, the goal of systematics is to have taxonomy actually reflect the evolutionary affinities or phylogeny of the organisms. So you essentially want everything to sort of work well together. So you want the taxonomy to actually show the phylogeny of organisms. So a phylogenetic tree is a branching diagram that shows evolutionary relationships between organisms. Here's an example of a tree, and so here we're looking at carnivores, and so that's the order. You can see we've got four different genus right here, and then we've got our different species. So these trees are going to show ancestral lineage, they will show branch points or nodes, and then they're going to show branches of different length, and the length of branch points is going to suggest the amount of time or the degree of closeness. So now a quick question is, how do you group taxa so that the phylogenetic relationships are correct? So the ideal situation is to have monophyletic grouping when a single ancestor gave rise to all the species in that taxon. So here is an example. If we were looking at a phylogenetic tree, here you have sort of this ancestral species B here, which gave rise to all of these species up at the top. But there are other possibilities as well. So polyphyletic grouping is where members are derived from two or more ancestral forms, and then paraphyletic is when grouping does not include all the members from an ancestral form. So here we have the polyphyletic grouping, and so we are looking at the ancestors of C and F, and paraphyletic here we are looking at K, but we are not including J. So the problem that we face with this is that not all likeness is inherited from a common ancestor. And we talked a little bit previously about homology versus analogy. And so homology is the likeness that's attributed to shared ancestry. And so the examples that we talked about before were the forelimbs, sorry about the typo there, of vertebrates. So like, especially when you look at mammals, we talked about how our hand and arm can look similar to the wing of a bat or the fin of a whale. And then you've got analogy, which is when you have likeness due to evolution, and it's because it's a solution for the same problem. So you've got insects and birds which both fly, so they both need wings. So even though they both have wings, the wings are designed very differently, and so that's an example of analogy. Convergent evolution is when you have unrelated species with similar adaptations to a common environment. So this is a very specific example of analogy. And so here we could look at sharks and dolphins. So just looking at sharks and dolphins, morphologically, they look very similar. But we know that dolphins are mammals and sharks are not. So they have very similar adaptations in some cases because they live in a common environment. Here we have another example where only one of the individuals you're looking at here is a mole. Here you've got plants, and only one of these is a cactus, even though they look very similar. So we need methods to group organisms by similarities and phylogeny. And so one way to do this is through molecular systematics. And this is when you're comparing similarities at the molecular level. So you're looking at DNA and proteins as opposed to just an overview, a morphological overview of individuals. And this allows us to look at different patterns within the DNA. So if you have really similar DNA, then you can assume that what you're looking at, the individuals or species you're looking at, are more closely related, and they have a more recent common ancestor. Whereas if you're looking at groups that have very different DNA, they're less closely related and a less recent common ancestor. So it's just another example. So again, we've got Drosophila, very, very far away from like the mouse and the human. Oops. So when we're making a phylogenetic tree, you can use morphology, you can use genetic data, or you could use other types of data. And we like to typically root them in a common ancestor, and then use statistical analysis looking for best fit. And what we mean by best fit is something called maximum parsimony, and that requires the fewest DNA-based changes. We can also look at branch lengths that suggest the closeness of the relationships and the time of the branch points. So evolutionary history is in the organism's genome. And so what that means is we can look at the number of base pair changes, comparing one type of organism to another. So just a quick note that taxonomic relationships can be changed based on what DNA tells us. So this is fairly new, looking at phylogeny using DNA. So prior to this kind of technology, we essentially were relying on a lot of morphological data to group things. But now that we have the DNA technology available, it can actually change some of these taxonomic relationships. The result is that taxonomy will become genealogies reflecting the organism's descent with modification. So we're going to talk really quickly about the systems that we use to organize species. So we've got kingdoms. This is the highest taxonomic category. The old system, you just had 2 kingdoms, plant and animal. In 1969, we had a 5-kingdom system. This has been changing. But the main characteristics that you were looking for were cell type, structure, nutrition mode. But we did have some problems with a couple of the kingdoms, and so now we have a 6-kingdom system, and so we're looking at archaebacteria, eubacteria, protista, fungi, plants, and animals. And then we also have domains, and so this is actually a classification that is higher than kingdom. So we have a 3-domain system, again based on molecular structure for evolutionary relationships. And we have this 3-domain system because not all prokaryotes are alike, and so we actually need to break them up into 2 groups. So you've got bacteria, which are prokaryotic, and then archaea, which are prokaryotic, but they are more similar to eukaryotic cells. And then you have the eukarya, which is the traditional eukaryotic cell. And this is just a diagram from your book that is comparing different characters of life between the 3 different domains. And this is just a breakdown showing the common ancestor of all life within the 3 domains. And so your summary, you guys need to know what is phylogeny, what is a phylogenetic tree, and then also how is molecular data used in phylogeny. And so for your lecture question today, I want you guys in your own words to compare phylogeny, systematics, and taxonomy. How are they different? How are they similar? So send me a message via the class website with your response. Thanks, guys.